lecture for zoology that's online. Okay, we're going to pick up where we left off. Prior to this, we were talking about evolution through natural selection, the 5 steps that are involved with that. We talked a little bit about what it was meant by fitness and ways that you can look at fitness and ways that you could measure it. Okay, so for selection, if you want to know if selection is occurring in a population, what you would do is you would go out and you would measure a frequency distribution for some character in the population. Typically, when you're looking to see if selection is occurring, you're going to look at usually one character. It makes it easier to determine if something's going on or if changes are due just to random chance. Okay, so in this example, what we're going to look at is snails again. Okay, the character that we're going to look at is the color of the shell. Okay, so you can go out into a population and you'll find light brown, dark brown, tan, white, and medium brown, different variations in the snail shell color. Now it's assumed that the coloration of the shell is due to the DNA that the organism possesses. So if you remember that diagram about the central dogma where you have the genotype, which is the DNA, then the RNA, then the protein with the phenotype. So what the organism looks like, its phenotype, is going to be dictated by its DNA or its genotype. Okay, so the assumption is that these characters are, the traits are heritable. Okay, so you go out at time 0, you measure the traits, the frequency of the traits in the population, you plot it. Okay, so this is your starting distribution. Okay, so to determine if selection's happening, you would go out in the population at a later time, and then you would count the traits again. You would measure the frequencies and compare them to your original distribution. Okay, and selection, if it's occurring, is described in 3 ways. Okay, so directional selection. Okay, in directional selection, the frequency of the traits will move in a particular direction. So in this case, the dotted line represents the original distribution. This line, this area of the graph, represents the current distribution. So there's been a shift in the frequency of the traits. Okay, so what you see is the traits are moving towards the darker end of the spectrum. So it's moving in one direction, and that's why we call it directional selection. Okay, stabilizing selection, the frequency of the traits is going to be pushed towards the center or the mean. So again, the dotted line represents the average at the beginning when you first measure those traits and plot the frequency. When you go back the second time, okay, there are more, or there's greater frequency of that average trait in the middle, and the extremes are kind of dropping out in frequency. Okay, so there's selection against these really, really light colors and these really, really dark colors, and there's a greater selection for the ones that are right in the middle, the average trait. Okay, disruptive selection, the frequency of the traits are pushed towards the ends or the tails of the distribution. Okay, so again, dotted line represents the initial frequency. Blue area under the curve represents the current frequency. So over time, the main trait or the mean trait of this light brown is selected against, and the extremes, so the lighter colored and the really, really darker colored traits are selected for. Typically, you'll see this kind of selection occurring in areas where you have a patchy distribution or a patchy habitat, where there are little patches of light colored or dark colored background or habitat. Okay, one way to kind of see that or visualize that is if you've ever been to the beach or the shore and you're at the water's edge and you move up along the sand. As you move further and further away from the water, it goes from sand, and then you start to get some patches of grass and some patches of little shrubs, and then eventually you move in and may become a forested area. So in that transition zone between the beach, the sandy area, and the wooded area, you have these patches. So there's some sandy areas, there's some vegetation areas, and that's where you tend to find this kind of selection, this disruptive selection occurring. Okay, shift gears a little bit, some terminology Taxonomy is just a classification of organisms. It's how you take organisms and put them into particular groupings. Okay, phylogeny, which we've done a lab on, we've talked about this a little bit. It's a evolutionary history of a species or a group of species. Okay, phylogenetic tree is the branch diagram. So those things that we constructed in the lab that we call phylogenies, technically they're phylogenetic trees. These two terms, phylogeny and phylogenetic tree, they're used interchangeably. Okay, this is an example of a phylogeny or a phylogenetic tree of the phylum Cnidaria. Okay, we're going to talk about this taxonomic group, this phylum. We're going to look at those in probably about a week in lecture. So you don't need to really worry about this right now. Okay, so just kind of going through some of the general constructs of phylogeny. Okay, you've got all these lines, you've got these branches, these forks, okay, and then you have organisms listed at the top, and you've got these characteristics kind of listed along the branches. Okay, so question for you, what do you remember about phylogenies? Okay, we're going to go through and talk about those things, but you want to kind of think in the back of your mind, when we made that phylogeny, the hardware, what are some of the things that we looked at? Okay, so when you construct these phylogenies, okay, there are a lot of ways you can do it. The most common way is to look at morphological characters. So you look at an organism and you look at the characters, the things that you can describe about it. Okay, and organisms that have more similar characters are tended to be grouped as families or on similar branches. So those characters that you look at can be described or categorized in two ways. There's primitive and derived. The primitive are features that haven't changed from the early ancestor, meaning the ancestor had it and the currently living species have it. Okay, derived characters are features that are unique to the individual groups. Okay, so you don't see them in the ancestor, you see them in the current species or in smaller branches. Okay, one way to remember the difference between the two, derived starts with D, different starts with D. So the derived characteristics are what make groups or branches different from everybody else on that phylogeny. Okay, so when we use those characteristics to construct the phylogenies, okay, the derived characters are the ones that are located closer to the top. Primitive, by definition, the ancestor had it, living species have it, so those tend to be found towards the bottom of the phylogeny. Okay, so let's see if we understand this. Okay, nematocysts, which are right here. Okay, so down here, nematocysts, would you say that's a primitive or a derived character? Okay, so primitive. It's a primitive character. It's towards the bottom of the phylogeny. Okay, so the ancestor had nematocysts. Everything above it has nematocysts as well. Okay, and what we're going to see is the cnidarians, that's one of the characteristics that separates them out from all the other animals, is they have these nematocysts. Okay, it's not in the slides, but I think it's in your outline notes. Strobilation. Okay, so here's strobilation up here. Would you consider that to be a primitive or a derived character? Okay, it's up towards the top, so that would make it derived. It's a derived characteristic. Okay, so derived is up towards the top, primitive down towards the bottom. Okay, extinct and extant. Extinct species, no longer living. There are no living representatives. Okay, they did exist. We know that through fossil record. Okay, extant species are species that are still alive. Okay, so something like crocodiles, alligators, and so on. Okay, so here's a phylogeny. Okay, it's a general phylogeny with these letters. Okay, which of these letters would be considered the extant species, which of these letters would be considered extinct? Okay, and this you probably may remember from when we did the phylogeny lab. Okay. Okay, so if you guessed or you thought that the extant species, the letters are at the top, at the tips of the branches, that's correct. Anything below is considered extinct. Now I have to apologize because this mouse, I'm touching the side of it and it's actually causing things to, flip back and forth. So I'm going to keep my hands off of that and I'll just do this kind of thing. Okay, so on our phylogenies again, okay, we have the branch points. Okay, they indicate common ancestry. Okay, so in this case, letter D, species D, or group D, group E, share a common ancestor. They're more closely related because they're on the same fork. Okay, this branch point, again, as we talked about when we did the lab, okay, indicates a common ancestor, and that's what these letters represent, the common ancestor. Okay, they're extinct because they're down below the tips. Okay, that fork or branch. Okay, so when we're constructing phylogenies, one of the things you have to watch out for is characteristics that you're describing in different organisms. Okay, so characteristics or structures that you look at can be described in one of two ways. One of those is analogous. Okay, analogous means it's like something. So analogous structures are characters that have similar structure and function, but different evolutionary origin. Okay, so what they do is they allow organisms to kind of accomplish the same thing, but how they got to that point where they can accomplish the same thing is different. Okay, so wings. Wings provide organisms the ability to fly, and that's powered flight. Okay, so the wings of a butterfly and the wings of a bird have the same endpoint, which is allowing the organism to fly, but how they came about is totally different. Okay, so if you look in the wings of a bird, you're going to find that there are bone structures that are similar to what we see in our arms. If you looked at the wings of a butterfly, you took a microscope and you kind of looked in here, would you see those little bone structures in there? No, you're not going to see that. Okay, you're going to see other chitinous structures, these hard structures that give support to the wing. Okay, so they're analogous, they're similar. Homologous, characters with similar function and structure, but they have a common evolutionary origin. Okay, so the limbs of vertebrates, okay, things with a backbone. So our arms, a human's arm, the wing of a bat, the flipper of a porpoise, leg of a horse. When you compare the bony structures inside of them, okay, they all pretty much have the same basic setup. Okay, so we can say with some confidence that these structures all came from a common ancestor, and that's what makes them homologous. Classification. Okay, with classification, I'm actually going to stop there. I'm going to do another lecture separate on classification because we're kind of at 15 minutes here. Okay, so next lecture is going to be the classification.